diseases emerged, and one large pandemic, which messed with our life for 2 years now. I will start the story about electron microscopy. At the beginning of 20th century, Victor Babeș came in Romania and developed an institute of bacteriology and pathology. He was one of the most important pathologists and bacteriologists at that time, and wrote the first textbook of bacteriology along with his friend Cornel. And he was interested in almost all infectious diseases, with specific interest in rabies, which was quite frequent at that time. He described some pathological lesions specific for that disease, and also applied therapy, anti-rabies therapy. Vaccine was settled by Pasteur 2 years later, and Babeș already started to use. Even the pathogen was not known at that time because the techniques was not available. The virus was supposed to be the pathogen because it was too small to be filtered. Was also non-replicative in culture media, and required cell culture to replicate. So we need to thanks to Ruška brothers for electron microscopy. In fact, Ernst Ruška designed the first electron microscope in 1931, and his brother was interested in viruses and bacteria, and asked for improvement in this first model, and Ernst did that. We have here the first paper showing you some viruses. Mousepox, in fact, is here. Looks like a nanoparticle you are working with. But meantime, the electron microscopy developed, and the technique and the software and the new added technologies of ice producing in a vitreous state improved a lot the electron microscopy. Why to use electron microscopy to identify an unknown pathogen? Because it's quite easy, quite fast. 10 minutes will be enough to see a viral particle using negative staining, a specific technique. You can see 2 or more different infectious agents at the same time in the sample, if is the case. Does not require a living organism. In fact, processing the samples for electron microscopy requires a fixation, and glutaraldehyde is killing almost everything. We know we need some reagents, of course, to increase the contrast in electron microscopy images, but not something special like antibodies or PCR products, nucleic acid standards, or and so on. We have no false positive results, and this is a good thing. And we can see, in fact, the infectious agent, and we can classify the pathogen. We have some limitation in electron microscopy. We need a quite large quantity of pathogen in the sample, but usually when it's a infectious disease with unknown pathogen, the lesion will be quite dramatic, and a lot of particle will be available in the biological sample. If it's not present, this is not equal with the absence of the pathogen. And we have some limitation linked with the expensive instrument, which is the electron microscope. And expensive knowledge which is not always available. We need a biologist or microbiologist trained to recognize the pathogens in this kind of techniques. Transmission electron microscopy is the best way to look for viruses mostly, and bacteria of course. And we have available some techniques from classical plastic embedding procedure, which is quite laborious and time consuming if we need to look for infectious agents in tissues or cells. To the quite easy to do negative staining technique where the particles will be bright on the dark background and easy to recognize. And new techniques which involve the specific equipment and a specific technique to obtain the vitreous eyes and to embed the particles inside. But the resolution is quite good, allowing us to see molecular details at atomic level. Negative staining is a technique which is quite easy and fast. In 10 minutes we can have the grid prepared for the microscope, and 20 minutes to examine the grid will allow you to put the diagnosis. This is the only support which could be introduced in an electron microscope, a transmission electron microscope. So we can put a little drop of biological fluid containing the particles. We can let the grid to dry, and after that we cover the particles which are on the grid with a small thin layer of metallic solution having large atoms. This solution will prefer to stay in lower places surrounding the particle and creating a dark background for the particle, which is quite shiny in this technique. Mostly phosphotoxic acid gets used for negative staining, and this technique was used for identifying almost all viruses in 60s. June Almeida is one of the researchers who was interested in viral classification, and he wrote a poem for this technique, negative staining. This is her image took in 1966, having a coronavirus unknown, zoonotic origin. But also she tried to classify the viruses according with their location into the cell, into the nucleus or into the cytoplasm, according with their size or their morphology. And this classification is still in use today. Even we have nice cartoons emphasizing the structure, the morphology, the different morphology of different types of viruses. Most of them are quite nasty for humans. All this information comes from electron microscopy. Negative staining or classical transmission electron microscopy, we have all these morphological details like size, shape, internal structure from electron microscopy. So the electron microscopy is quite a good technique to discover the new infectious agents. In the last 10 years, maybe, the electron microscopy changed a little bit. We have a new type of electron microscopy, cryo-electron microscopy. The liquid nitrogen temperature is the temperature which is quite stable inside the microscope, and we can see viral particles or any kind of molecules frozen in a thin layer of vitreous ice in a state most appropriate from native states. So we have no need to put different heavy metals or chemicals to have a contrast, because we have this technique which is sustained with quite sophisticated softwares, which allow us to have a nice image of particles, molecules. In this case, influenza virus is quite visible, trapped in the vital signs. This kind of technique is used today to detail at the molecular level, at the atomic level, all the molecules composing the viral particles, and we have some databases like this one, protein data bank, which is holding the informations acquired through cryo-electron microscopy, and electron microscopy data bank, also a repository for data obtained from cryo-electron microscopy and cryo-electron tomography, in fact, because we can do tomography. We have 3 types of microscopes in Victor Babeș Institute. The institute built for Victor Babeș, in fact, when he came in Romania. We have a small one they use for diagnosis, for mostly for kidney biopsies diagnosis, and 2 other more complicated, and one with cryo facility, cryo capability. Our experience is not quite large with viruses or infectious diseases, mostly with kidney diseases. We work with kidney diseases, and sometimes we have kidney transplant biopsies, and transplant biopsies involve some immunosuppression, and these patients have an immune system compromised, and sometimes infectious agents would be developing in their kidney. Even quite benign polyomavirus, which can build some large nuclear inclusion, like this one here. If we enlarge the image, we can see the particular structure, and even closer, we can imagine, mostly in this transmission electron microscopy, the ecohedral symmetry of the virus. In the transmission electron microscopy sections, when we do the sections from plastic embedded tissue, the section is about 100 nanometer thick. So we have the entire particle in the section, and even more, and sometimes we have different layers superimposed in the same 2-dimensional image. But in this kind of technique, we can see viral infection, and also associated with bacterial infection. And sometimes this diagnosis could be unresolved in clinical laboratory. Another experience we have with viruses is the negative staining of different inactivated influenza viruses for vaccine producing protocols. So we can do negative staining and measure the particles which are changed by different experimental conditions, and the morphology, the changed morphology could give us some important data. This experience was before we had this electron microscope with cryo facility, and we resumed the collaboration with our colleagues from Cantacuzino Institute for vaccine producing with cryo-electron microscopy. In some conditions, it was supposed to have a change in a particular matrix protein in influenza virus, and this modification of M1 protein, it was suspected to generate a quite fragile membrane for influenza virus vaccine. And we try to see in cryo-electron microscopy if the changing solution for inactivation viral particles will change the structure of the membrane. So we measure the membrane of the influenza viruses in different conditions, and this is in cryo-electron microscopy. One big problem for that is that influenza virus is quite polymorph. We have small particles here and larger particles in the same solution, in the same sample, and the membrane could be different in bidimensional images. So we decided to do electron tomography, cryo-electron tomography. This involved to tilt the grid inside the microscope and acquire serial images, reconstruct a volume. And this is the tomography, one of the tomographies, and you can see entire particle in the volume reconstructed. From this reconstructed volume, we can extract some digital sections which are very, very thin and could measure the membrane. From tens of cryo-electron tomograms, we extract some particles and made some libraries containing thousands of particles, and measure the membrane in different conditions, in different experimental conditions. And for sadly, we have no good result. The two conditions we had investigated didn't change the membrane, the structure of the spike protein of the or the M protein, matrix protein, and this happened in 2020, and everything changed because at that time a new pandemic outbreak with coronavirus 2 emerged, and our colleagues in Cantacuzino Institute were diverted in different other tasks. This is the first image of coronavirus 2, SARS-CoV-2. We have clear spikes around the central particle, and this image directed the research to the coronavirus to be a culprit for the pandemic 2020. After this image, of course, all the molecular techniques Professor Flores already presented you started to be used to generate a test, a specific test, sequencing the genomic material, give us the PCR test, and so on. So some coronaviruses were culprit for endemic outbreaks of infectious diseases and this pandemic in 2020. We have here a negative staining images of various coronaviruses, SARS-CoV-2, SARS-CoV-1 in 2003, and a transmission electron microscopy showing you particles inside a cell. In transmission electron microscopy for plastic sections, the corona glycoproteins will not be visible because of due to the processing of the cells for transmission electron microscopy. Cryo-electron microscopy was useful studying SARS-CoV-2 virus because detailing the atomic structure of the spike protein was important to predict the variant shifting or to decide if new antibody designed specific for specific three-dimensional structure of the spike protein will be effective in the therapy. We had a vaccine quite quickly for this infection disease for SARS-CoV-2, and we did a negative stain for the two types of vaccines available today. The classic one with TAN adenovirus, very nice particles visible in this image, and for the new type with synthetic RNA in a lipidic shell. They are looking very nice, very clean, very beautiful, and for sure electron microscopy would answer this question without no problem. No, the COVID vaccine cannot connect to the internet to anybody. We made a small experiment giving a vaccine in cell culture to a certain type of cells, and we observed the specific, the classic route of internalizing the vaccine to endocytosis and endolysosomal compartment specific was filled with this ribonucleic particles. We observed after 12 hours that some very few cells exposed on the surface. This image here is a magnification from this one. Few cells exposed on the surface, some spike proteins, which could be in a living body exposed to the immune system and trained to generate antibodies. So this kind of extension, cellular extension, is quite strange and not usually seen in cell culture, but you can see here very nice spike proteins in cell after 12 hours. This is a team of my lab, electron microscopy lab. Emmanuel is the one who performed the testing of cells in culture with vaccine administration, and he tried to reconstruct the spike proteins from the cryo-electron microscopy images. We worked in collaboration with Professor Onu and his team for influenza vaccine, and we have a lot of students from medical faculty to working with us for image processing, especially because it's time consuming and require quite a good knowledge of the software, so they are interested in this kind of things. So this is the story I want to tell you about the electron microscopy as a useful tool for diagnosis of unknown infectious agents, mainly viruses, but also I believe will be a good tool for the study of bacteria and their viruses, fungi, because the microbiota it's a new topic in medical field, and we know very few things about this type of cell in our body. Thank you.
Thank you very much. We have time for a question. Okay, and now our last invited speaker. We are honored to have here Otto Larsson with inverse problems and hierarchical multi-scale modeling of biological matter.
Yeah,
Welcome.
that's correct. Which one? Oh, this one. Okay. Okay, thank you. First of all, I would like to thank the organizers and Professor Simeonescu for inviting me here. It's a great pleasure to be here. I'm going to tell you a story which is quite different from what you have heard. Today it's about in silico studies, and this title is basically also the outline of my talk. So I will first tell you something about inverse problems and how to solve them. Then I will focus on hierarchies in biomolecular structures, and then I will introduce a method we developed in our group, which is quite general, but I show examples on how to use it to study biological matter. I should of course say I come from JAS, Intel Centro. I'm in the group of Dr. Maria Pitala, and I just want to say that I really enjoy to be here. There is an incredible atmosphere, scientific atmosphere, interdisciplinary, and I have learned a lot in working there. Otherwise, I come from Stockholm University. I did graduate there, and I also did all my scientific career, except all the breaks I did abroad. And also now, once in a while, I go to Nanjing Tech University. I'm a member of staff there with some teaching and supervision, and also I'm a visiting professor in the most northern university in Sweden, Luleå. It's very close to polar circle, and this picture is taken in the middle of December, where there is basically no daylight, but you can see very often beautiful northern lights there instead. Okay, so about inverse problems, actually you all, what I've been hearing today, when you do imaging, you solve inverse problems, you don't reflect so much about it, but otherwise, if we stop thinking about it, so when we solve inverse problems, we invert what we know, find something we don't know, and then this way a solution is the inverse of the problem. So normally we solve forward problems, so here is a very simple equation. Actually it doesn't need to be the linear line crossing the origin. X can be some very multifunctional equation of the data structure, and K is an operator. So finding the inverse, solving the inverse problem, then we have to find some kind of inverse of this operator. But to solve inverse problems uniquely is typically very difficult, and this depends on inconsistencies and noise in the available data, and also the parameter space is often very, very large. You have to explore. But we solve continuously inverse problem. Every day we are trying to understand what just happened and why. And of course we use common sense and all life experience to try to figure out that. In science, we very often use two things which are not so easy to define: educated guess or reverse engineering. So this lady is just studying a test tube after a synthesis, trying to find out what did I get here actually, and then you can do reverse engineering, and modeling is often very useful in reverse engineering. Okay, in nature, all living species, birds, snakes, whatever, they are solving inverse problems to find food and also to survive. They use, they rely on perceptions, like sounds and light and smells and so on. In science and mathematics, solving real inverse problems is common. There are scientific journals for this purpose. And currently it has become a kind of hot topic because it's boosted very much by machine learning and big data. I'm sorry, I've been watching this TV series, Crimes in Investigation, and it's about solving forensic inverse problems because they have to reverse the time to this point of time when the crime did happen, and sometimes you get really impressed when you watch this TV series, how they use all kinds of science from biology to whatever, and they immediately make very fast conclusions. And so, then, yeah, solving inverse problems can also save your life. No. Okay, so about hierarchy. Nature prefers hierarchy in structures it develops. Nature is the most superior material scientist there is. And one good example is the genome, starting from DNA and how it is packed in nucleus and core particles and chromatin and so on. And this first structures that are very well known today and studied, and also the chromosome, but then there's a gray area here which we still don't know so much about because we have to take into account the dynamics also, and also at different points of the cell cycle, different things happen there. So I will come back to this a little bit later in presenting you some multi-scale modeling, which is the topic of my talk. Nature also prefers composites together with hierarchy because it's this way you can nature can create very strong structures easily, and then these structures can be expanded and they can grow and so on. This is a piece of our skin, which also can be seen as as a hierarchical structure, and it can be modeled as well. So all these structures are really ideal for hierarchical multiscale modeling. So now I should say something about multiscale modeling. But first, modeling is also called in silico studies. It's computations, it's studies taking place in a silicon chip, and so on. And when you do in silico studies on biological structures and processes, there are very, very many challenges there. And one is, of course, the incredible number of orders of magnitude in length and time scales and other scales. And then finally, it's the biology itself, which is built successively in the matter. And it requires very specific conditions, so it's very complex, diverse, and there's so much chemistry and energetics and thermodynamics, etc., etc. And finally, in silico studies, it's completely beyond reach life itself, self-organization, and so on. But we can try, at least on this point, to do modeling. So if you look at the so-called nanoscale, it starts perhaps from 1 angstrom and it goes to 100 micrometers. It turns out that when you do in silico, there is no single modeling method you can use to cover all this nanoscale. Typically, you can start with so-called first principles methods, quantum mechanics, quantum chemistry, where you create atoms and molecules from nuclei and electrons. Beyond that, you go to classical mechanics. You use something called molecular mechanics, which doesn't contain any more electrons, but you have atoms, and from atoms you build molecules and molecular systems, liquids and solids, and so on. So you wonder where these limits come from. They are actually only practical limits given by the computers and the computing power we have at the moment. So it means that the limits are moving slowly to the right when computers are becoming faster and faster. After that, we have soft matter and mesoscale, and we can forget about the individual atoms and molecules, and we treat aggregates of them. And we can study soft matter. We can also study organelles, and well, chromosome was one of the examples, and different types of complex biological soft matter. So here are a few examples of these structures. I will come back to them later. So now I introduce this method which we are using, which we developed actually a long time ago. And it is based on something called successive co-straining. And in every co-strain step, we solve this so-called inverse problem. So the entire method is called the method of inverse Monte Carlo. It actually combines very well with something called multiscale imaging, because you can do imaging also at different length scales and so on. So they combine very well together. So let's start from these first principles simulations. I will not explain these methods. So this means Born-Oppenheimer molecular dynamics. This means Carr-Parino molecular dynamics. This means tight binding molecular dynamics. They're not they're not important here when I describe this this method. But they are methods to perform simulations to treat molecules and how molecules move and their dynamics. And as we saw in the previous slide, we cannot use these first principles methods for very large systems, so we have to start with limited size of systems. So we do a simulation, and now from results from these simulations, we solve the inverse problem. We create a model which we can use on the next level. But when we use this model, we get exactly back the same results we got in this original simulation. Doing this, we throw away all the degrees of freedom we don't need in this next level. And going from quantum mechanics to classical mechanics, we don't have much use for the electrons, so we throw them away. Now, here we can increase the size of the system like 2 orders of magnitude because we don't need to treat with electron degrees of freedom. And we do exactly the same. From results we get from here, we can create a model we can use again on the next level, which is the mesoscale. And here we throw away lots of things which we don't need here. we throw away the atoms, we throw away the bonds, we throw away, for example, solvent entirely. So we just have particles there. And this way we can go up several levels. We can co-scale the co-scale thing, create a new model, go to the next level, create a new model, and so on. And this is the level many of the methods, experimental methods we heard today, also come from top down. So this is obviously from bottom up approach. And then what we can do is we can also go back. This is called fine graining. We have to try to reinvent these degrees of freedom we did throw away. It's not trivial at all, but it can be done to certain systems. If we want to go further up, we have to abandon the particle description of the matter, and then we can use elastic networks, finite elements, lattice Boltzmann, and other types of methods which are not based on particles anymore. But this methodology here, it can be used, for example, in non-equilibrium simulations, like for red blood cells flowing in the capillaries. It can be used for drug transport and delivery. Cellular uptake can be modeled with these models and many more applications. So this experimental techniques also, we saw many of these videos today with particles moving slowly around. They can be used to create these soft matter particles, and also the interactions can be estimated from that. And then this can be used in simulations further. Okay, so This inverse multivariate method, it provides something called solvent-mediated interaction pair potentials. That's a force field. That's something everybody who has done molecular simulation knows what force field is. So possibly not all of you know, but it's like an interaction law how particles, when they come close to each other, how they feel each other, and what type of forces they use to against each other. And so we use structural information. We can take it either from experiments or also from very accurate theoretical simulations. And the secret is that there is a correspondence between something called radial distribution functions, GOR, and these pair potentials, this force field. And historically, we can go back early 60s when already the first time these GORs were inverted and potential mean forces of them were calculated, potentials for liquid metals. The real breakthrough came in the mid-70s when Henderson did prove a theory that there actually is a uniqueness between radial distribution functions and pair potentials. This means that when we solve this inverse problem, we can solve it uniquely. He also showed later that it is true for multi-component systems. However, this theorem doesn't say how to obtain one from the other, so this has to be invented. So our inverse multical is based on something called Svensson's multical renormalization method on Ising models from late 70s. There are also other similar structure-based methods, and I would like to mention one from Florian Müller-Blatte. It's called iterative Boltzmann inversion. In fact, we use it always initially because it's a very simple method, and then we go at some point we go to our own method to get better convergence. Okay, so it's based on structure, this, and I like very much water molecules. They are among the simplest molecules there are, but they are also the most complicated molecules. And here is a group of water molecules. You probably can see it, and they're very happy. They decide let's take a selfie or a groupie, and then one water molecule then keeps this stick with the camera, and this camera has its shutter open all the time. That means that the images are overlapping, they come on top of each other. And when you look at this bunch of water molecules, it also applies for all liquids. Liquids are very chaotic structurally, but there is still a structure there which we don't immediately see. But now suddenly we can see the structure in this groupie photograph. And it is because when this one particular water molecule keeps this camera, this stick, he also creates a molecular coordinate system on itself. So it means that this water molecule stands still, whereas all the other molecules are moving. And this picture also shows that this water molecule donates two hydrogens to these other water molecules, hydrogen bonds, and also it accepts two. So this could be already the structural information we use, but we do something else. We use these radial distribution functions, which actually is the same thing, but not instead of in the molecular coordinate system. Now we calculate the distances and interactions in a laboratory coordinate system. So we get the function like this. There's a first maximum, there's a second maximum, and this is the function we invert to this pair potential. Radial distribution gives the probability to find another atom close by at a certain distance, and then it is normalized to the bulk density. And this function can be obtained from molecular simulations or also from diffraction studies, electron or neutron diffraction. But you will get it as a structure factor in a K space, which you have to Fourier transform to real space. And then first. Okay, so this, I think I will skip it. It describes a little bit the theory about this inversion, but what we use is Newton-Raphson. We start from the reference, the accurate radial distribution function from simulation, for example. We calculate potential and mean force, and we use it as a potential to calculate the radial distribution functions. And in the first iteration, yeah, I would say the black curve here is the reference, the exact. But the first iteration, we get something which is completely off. But you see, after 10 iterations, it converts to the original radial distribution function. And when this happens, we get a coarse-grained potential function which we can use. And this we can use at all these levels in this ladder you saw previously, and we can calculate the potentials. So I give you some examples here. Here is water in water. So you see here is the OO distances, but there is also OH and HH. And when you invert this, you get pair potentials. These are actually not coarse-grained potentials because it is exactly like a new water model. But if you study like sodium chloride, table salt in water, you have the contact peak for sodium and chloride, you have the solvent separated and so on. And when you invert this, you get potentials which look like that. They don't look like Lennard-Jones or any simple potentials. They are quite ugly and they oscillate. But that is because they contain the hydration shells. So we can use these potentials without water to do the simulation, and in a few seconds we get these radial distribution functions which took several weeks when we had all atom potentials there. Okay, so we can go to more complicated systems like phospholipids. We invert selected radial distribution functions, we get effective potentials, which we can use in coarse-grained models, and so on. If also you notice that at long distances, these potentials, they oscillate first, but then they coincide with Coulomb's law. So this makes that you can divide these potentials to short-range parts and a long-range part. And of course, the short-range part is very ugly, wiggling functions, whereas the Coulomb's law is very smooth. If you use this method to simulate, for example, DNA, you have to decide the topology and how do you coarse grain it and so on. So this is an all atom description of DNA, and this is a very simple coarse-grained model. This is where there is one nucleotide per bead. Here we have sugar, phosphate, and base as beads, but then we have to decide topology. Then you can use it in simulations like this, where we have studied up to 500 base pairs, twist and drift of DNA, mini circles, and so on, which you probably have seen in old-fashioned telephone. However, this is which shows how useful this method can be. So, if we start from a system where we have all the atoms there, including water molecules and the counter ions around DNA, when we coarse-grain this, in the next step, you see there are no water molecules or ions anymore, because now they are in this effective coarse-grained potential. We can then use this to create nucleus and core particles. Here, actually, the interior is made of elastic network model. And then you successively coarse-grain it and make the system bigger and bigger. And this is one piece of this chromatin, and it contains only 98 beads. And if you go back and describe this on all atom level, you would need 11 million of atoms to describe exactly the same piece. And this way we come quite long in describing the genome. What is left here is this gray area again, which if we want to go further, we should use a very very simple, let's say, polymer models or something, but then we should combine it together with experimental methods like fish and Pisces and so on, and then we can go further. So this is something we are working on at the moment. So this should be a very good example of systematic hierarchical quantification modeling from DNA to chromatin based on solving immense problems. So this is my last slide also. So thank you very much.
Thank you very much. It was really invigorating. Yes. So we'll try to give attention possible. So we have some questions.
Yes.
Please.
Thank you. It was a very nice presentation. I enjoyed it. I'll highly read your paper. I didn't respond. I didn't know about it, so I should. The problem I would like to ask you, so the method of getting from the pair correlation functions back to their potential is working only if I have spherical symmetry for the pair correlation, no? So you, no, sorry, for the potential. So usually the two-particle potential, what you are looking for, it has also direction dependence, and then the Euler's problem should not work. Yeah, going from the pair correlation function back to the potential, no?
Yes. Well. So what we do, we create the asymmetry what you're referring to.
Yeah, there is a direction dependence on the potential.
We don't actually have it because we have, like in atomistic simulations, we have pair potentials. So the pair potentials are between two particles, and using enough different pair potentials, you also describe this is asymmetry or whatever. But within these pair potentials, there is uniqueness between between pair correlates and
Okay, so
functions.
if you consider only two particle interactions and not the three,
Yeah.
then it is working.
Yeah. But of course there are other problems which I didn't mention, and because it is based on structural information, so you should not expect that you get dynamical properties correct, and because it gives two soft potentials, it also gives two fast dynamics. But if you use these potentials in Langevin dynamics, then you can choose the friction coefficient. So you calibrate it with experiments,
And also the noise, you can take it out, the noise and the noise properties from it. Because in the Langevin equation you have also the noise, not only the friction.
yeah, sure. But of course, it's not supposed to be that accurate as all atom simulations. But when you start, you can actually start from first principle simulation, and that means that no empirical information goes in. But of course, we start from all atom level using some force field, and then this way, when we go step by step, we take with us the atomistic information. So for example, this Martini force field model with your Lennarton's potentials, it doesn't have these details as we have in our model.
So why do you call it Monte Carlo? Because in your scheme was nothing random, is there? It was totally deterministic.
Actually, in a paper we call it reverse Monte Carlo, and then we noticed a couple of months later that there was already a reverse Monte Carlo from Hungarian colleague. And then we started to call it inverse Monte Carlo. Then now we call it actually Newtonian inverse. So I should have used that. Okay. More questions?
Other questions? Thank you for your wonderful presentation.
Unfortunately, we have just 20 minutes, 25 minutes for the round table. The bus will come in about 20 minutes. So indeed, right. Most likely the start now, but we have the round table at the table.
Would you like to comment, put some subjects on the table? Or?
Well, I think I'm the last one to put any subject on the table.
Okay.
What I could do is to ask some questions.
Okay.
For instance, I was excited by all the presentations, and I may put some questions from a naive perspective. For instance, because one of the challenges that are faced by medical doctors, not by the physicists, for instance, what is how to manage persistence. So these very rare type of cells which manage to escape antibiotics. Do, for instance, PCR molecular methods, are they able to reveal the presence of such capability of such this type of bacteria, for instance, or flow cytometry? Is it possible? Are they able? Do they have the sensitivity for pinpointing these type of cells? another type of question, so most likely I am just asking questions, most likely we'll discuss the dessert or something like that related to this. I was also trying to understand why most of the results related to the amount of the bacteria we have, if there is an amount which is below 10 to 4th bacteria per mil, then it is stated as negative. And then I understood, or I thought that this could be related to the capability of normal people, let's say, of their immune system to stem such amount of bacteria. And if this is the case, then it has to be a relationship between the susceptibility to a specific antibiotic and the capability of the immune system of a infected person to respond to infection. And if this is correctly put in place, then it has to be another type of analysis revealing exactly this, so the capability of the immune system to react. Is there such an analysis usually performed? Questions? If not, how to relate this, let's say, diminished capability of the immune system to another threshold, 10 to whatever, 3, I don't know. So, as a physicist, I can ask questions.